Today, our group is going to be talking about mammoth mice, also known as the woolly mouse. So, it is a genetically modified mouse with traits inspired by the mammoth species. Before we talk about woolly mouse, we need to talk about woolly mammoths. So, they are an extinct species that roamed the cold tundra regions approximately 300,000 years ago. And the extinction of the woolly mammoth is curious to be caused by loss of habitat, climate change, and also hunting by humans. On the other hand, Asian elephants may have survived since they are more resilient to climate change. And this has sparked an interest in the de-extinction efforts of the woolly mammoth. Using advanced technologies such as genome sequencing and genetic manipulation, it has given hope for the revival of species, or at least to restore some of the genetic traits. Due to difficulties in using elephants as the model organism, scientists have opted to use mice as a surrogate model in gene testing, creating what they call woolly mice. So the creation of woolly mice is part of the de-extinction effort with the intention of producing an elephant cull with mammoth-like traits that will allow them to survive in colder climates. So the aim of this study is to determine and confirm the viability and expression of woolly mammoth genes in mice, as well as creating the foundation in the revival of woolly mammoth at the same time to restore the ecosystem and mitigate climate change. However, there's so many unknowns, new things, and possible issues regarding the study, and the main of it is ethical concerns, especially regarding animal welfare and the complication that may arise from manipulating extinct genomes. Since mammoth genes may behave differently, particularly in their regulatory functions, often missing in ancient DNA. And some are questioning if the resources used in this de-extinction effort might have been better utilized for conservation efforts of currently endangered species instead.
Now, let's dive into the method to generate mammoth mice. Here is the flowchart of generating mammoth mice by using multiplex CRISPR-Cas9 editing in mouse embryonic stem cell. The first step starts with Cas9 RNP editing to introduce targeted gene modification into mouse embryonic stem cells. These cells were electroporated with Cas9 protein, guide RNAs, and single-stranded DNA templates, also known as SSODNAs, for precise knock-in edits. After 48 hours, the cells are harvested for genotyping by signal sequencing. In parallel, we also single-sorted the bulk population of transfected mouse embryonic stem cells into a 96 wild plates containing the IMEF feeders for colonial expansion. Then we will carry out guide and SSODNA screening. For each gene target, we tested multiple guides, ranging from 1 to 7 per gene. Each bar represents the editing outcome for a specific guide or guide SSODNA combinations. Gray bars indicate total modification rates, while purple and green represent knockout and knock-in efficiency, respectively. The orange bar highlights the guide design that was selected for our main mouse embryonic stem cell experiment based on performance. Next is a sequential editing strategy used to generate multiplex edited monoclonal mouse embryonic stem cell line. In experiment D, we performed knockout of the FGF5, MC1R, and FABP2, and a knock-in of KRT25. While in experiment E, we knock out FGF5, MC1R, and TGFA, and knock-in of KRT27. The bar graph showing the knockout percentage in purple and knock-in percentage in green for selected colonial lines generated in experiment D and E. We collected cells from laminin-coated plates to ensure that no IMEF contamination during genotyping by signal sequencing. Editing efficiency were also confirmed by next-generation sequencing. After that, the bio-nanocircles plot was used to summarize the structural variation in experiment D and E monoclonal mouse embryonic stem cell line. Translocation will be depicted as the curved line across the center of the plot, though none were detected in either sample. The number of variants are summarized on the right. Then, the multiplex CRISPR-Cas9 edited mouse embryonic stem cells are used to generate high-yield woolly mouse chimeras. Next, the workflow to generate the gene-edited mice from our previously engineered monoclonal mouse embryonic stem cell is shown in the diagram above. We started by culturing the edited stem cell in 2I media, which helps maintaining pluripotency. Then we injected 8 mouse embryonic stem cells into 8 cell stage embryos from the C57BL 6N strain. These embryos were cultured to the blastocyst stage, then transferred into the surrogate mothers. After birth, we wait for 21 days and then collect ear punches for genotyping. This allowed us to confirm the transmissional edits from the stem cell into a developing embryo. Next, the diagram shows the image blastocyst stage embryos injected at the 8 cell stage with the mouse embryonic stem cell taken at 20 times bright field. On the left, we have a bright field image, and on the right, with the same embryo expressing the green fluorescence spot, confirming that the injected stem cells successfully contributed to the developing embryos. In the diagram, the knockout percentage and the knock-in percentage editing profile of mice generated from experiment D and E monoclonal cell lines are shown. Each column is the NGS data from n equals to 1 mouse. So the mouse with the number is the name of the mouse used in this study. Then analysis by the graph showing high total modification, knockout and knock-in percentage of each genes, confirming successful and consistent editing in both cases. The photos above shows the mouse 574 from the experiment D and mouse 571 from experiment E at 3 to 4 weeks old and 6 to 7 weeks old respectively. Multi-reference photos are from an age-matched strain mouse. Both mice 574 and 571 displaying a distinct woolly phenotype with lighter, thicker and curlier fur at both 3 to 4 and 6 to 7 weeks of age. These results demonstrate successful expression of the engineered hair traits directly in the founder generation. In this experiment, out of 90 embryos injected, 7 mice with 4 intended changes were born.
Now let's talk about the result. The resulting mice had multiple gene edits that recreate key woolly mammoth traits. First, all the mice had a loss of function edit in the FGF5 gene, which extended their hair growth cycle and caused hair loss up to 3 times longer than wild-type mice. They also had loss of function edits in genes like FAM83G, FZD6 and TGM3, which impact the hair follicle development and resulting in woolly hair texture, wavy coats and curl whiskers. Additionally, non-functional TGFA and a mutation in KRT27 contributed to the wavy hair phenotype very similar to that of the woolly mammal. For coat color, the mice had a modified version of the MC1R gene, reducing melanin production and giving them the golden brown fur. Finally, a truncated version of the FABP2 gene involved in lipid metabolism was introduced, which is another trait found in mammals. When comparing the mice side by side, you can clearly see the engineered mice had thicker, longer and curlier fur. Their coats are visibly golden brown, resembling mammal hair preserved in fossils. Next, the researchers aim to assess the success of gene editing in mouse embryos by using fluorescence as a marker. First, embryos were sorted manually by the presence plus or absence minus of the red fluorescence. As shown in the bar graph in figure D, genotyping confirmed that embryos positive for red fluorescence and scarlet 3+ exhibited high levels of editing across multiple genes, seen by the tall purple and gray bars indicating knockout efficiency and total modifications. In contrast, embryos without fluorescence and scarlet 3- showed almost no detectable editing activity. Moving on to the next set of figures, after implantation, a mice edited with early stop codons in genes such as FGF5, FAM83G, FZD6, and TGM3 developed a striking hair phenotype. The heat map in figure E summarizes the knockout efficiency across different genes and mice. Fresh and thawed refer to whether the embryo was used immediately or after freezing. We can see consistently high editing rates across conditions, particularly in genes related to hair development. Figure F further supports this by breaking down the efficiency per mouse line with very high modification percentage. The pie chart in figure G shows whole genome sequencing off-target analysis for mouse 520, confirming that most off-target effects occur in non-coding or intergenic regions, suggesting minimal risk of functional disruption. Finally, figure H presents the phenotype results. Compared to the wild-type mice, edited mice like 520, 523, and 525 exhibited significantly exaggerated, curly and dense hair growth, both at 3 to 4 weeks and even more dramatically at 6 to 7 weeks. Together, these results show strong genotype-phenotype correlation. Successful multiplex editing in embryos led to visible phenotypic changes in adult mice, validating the efficiency and specificity of the editing approach. That's all from me. Thank you.
Overall, based on the result, researchers have successfully used CRISPR-Cas9 to edit multiple genes at once inside the mouse embryo. The first gene is the FGF5 gene. Mutation in this gene has caused the mice to have a longer hair trace, resembling the adaptation to the colder climate regions, protecting them against the extreme temperature conditions. Next is the KRT27 and TGFA genes. Mutation in these genes has resulted in the wavy or curly hair structure in the mice, instead of the straight hair to mimic woolly mammoths for thermoregulation purposes. Next is FABP2 genes that is related to the lipid metabolism that may indicate the shift to the metabolism that is beneficial in the colder environment and for fat and lipid storage. Next is the MC1R genes that is responsible for the melanin production. The mutation in these genes has causes the mice to have a golden hair color instead of a normal grey color. Another technology that is being used is the RNAP-mediated knockout that can reduce the off-target effects. Next is the multiplex precision genome editing that can edit multiple genes at once, which is the hair color, hair texture. And next is the precision homology-directed repair that can precisely insert the key mammoth genes inside the mice, which is very efficient and minimize mistakes. This is very important since editing 1 or 2 genes is not enough to create a living co-adaptive like mammoth, since it needs a wider range of editing across the genomes for its survival and functionality like the real mammoth. Transgenic application by using a CRISPR-Cas9 technique to insert woolly mammoth gene inside a living animal cell showcase a precise gene editing. This approach is more specific and efficient compared to the older genetic engineering methods such as viral vectors or random insertion. It allows us to integrate desired traits such as cold-resistant traits that focuses on hair, fat metabolism, and blood proteins. This is an improved technique in terms of controllability and off-target effects, but it is not guaranteed safe yet. This transgenic application avoids full cloning since it focuses more on a gene functionality in the living cells and a safer step for the extinction species to create more animals that can adapt to the world's tundra.
I will discuss on the implications of this study. First, this research can help in de-extinction, which is bringing back extinct animals like the woolly mammoth by mixing their genes with living animals like elephants. It also gives us a way to study on how ancient traits work into this world. For example, by putting mammoth genes that control fat storage into elephant cells, we can learn on how mammoths survive in freezing winters. In terms of biomedicine, these genes could teach us about fat metabolism, thermoregulation, and even help in fighting problems like obesity or help humans adapt to climate change. In conservation, it might help create climate-resilient animals to restore whole ecosystem like in the Arctic. But there are some big limitations. Just because a mammoth gene works in an elephant cell doesn't mean it will work the same way in a real mammoth. The genomes are different, so the gene might behave differently. There are also risks of off-target effects, which is editing genes can cause unexpected problems like bad changes in cells, poor health, or strange behaviors. And beyond that, many scientists worry that de-extinction could waste money and distract from saving animals that are already endangered. In short, this study is exciting and full of possibilities, but it also reminds us to be very careful, both scientifically and ethically. Thank you.
All in all, in this research, scientists aim to explore the genetic foundations of co-adaptation in extinct woolly mammoth by transferring key mammoth genes into living mice, successfully creating genetically modified organisms referred to as mammoth mice. Using CRISPR-Cas9 gene editing technology, a set of genes from the mammoth genome, which were believed to be crucial for survival in cold climates, was selected. These genes were associated with some vital physiological traits such as dense and insulating fur, fat metabolism, hair color, as well as hair structure. The selected mammoth-specific DNA sequences were precisely edited into the genomes of mouse embryos. Following successful implantation and development, the resulting mice were studied for phenotypic changes. The genetically modified mice exhibited significant differences compared to the unaltered ones. For instance, they grew visibly thicker and longer fur, wavy hair texture, developed outer fat distribution that is typically advantageous in cold environments, and strong metabolic shift that lead to enhanced thermoregulation. Some technologies, including RNP-mediated knockout, multiplex precision genome editing, and precision homology-directed repair editing, were used that resulted in phenotypes that resemble the woolly mammoth traits. Importantly, this study represents an advancement in the field of de-extinction and synthetic biology, indicating that it is possible to functionally revive certain traits of extinct species even without bringing the entire organism back to life. However, this research also raises serious ethical, ecological, and practical concerns. Introducing ancient traits into modern species could have unforeseen consequences on ecosystem, animal welfare, and evolutionary trajectories. Looking forward, this work is expanded by editing a broader set of mammoth genes into larger, more physiologically similar animals, such as elephants. Scientists are working to create co-adapted hybrid species, which are elephants with mammoth-like traits. Future research will focus on making gene editing safer and more precise, and understanding how genes help animals adapt to cold. This research marked a new era where the lines between the past and present become less distinct. That's all from us. Thanks for watching.